Hi, everyone. My name is Kirthana Srinivasan. I am here on behalf of the Center for Biological Data Science, representing Virginia Commonwealth University. I'm here to share my research conducted under PI Dr. Brett Boyd. The human louse microbiome, insights into insect-bacterial coevolution and consequences for vector capacity. What are lice? Pediculus humanus, the body louse, is an ectoparasite that only feeds on humans. Areas of poor hygiene and crowding, such as homeless shelters or refugee camps, are at a significant risk for drastic consequences of body lice. Body lice appear to present a significant threat to public health as they are competent vectors of disease, while head lice are poor vectors. In our review, we carefully examine microbial associates of human lice and identify evidence of louse-bacterial coevolution and its host respond to symbiont activity. What is a symbiont? So a symbiont is not just any gut bacteria, such as you or I might have. A symbiont is a specialized organelle that only thrives within the human louse microbiome, which means this endosymbiont cannot survive outside of the human louse microbiome. The interaction of the host-symbiont relationships is mapped in the figure above and talks about how this endosymbiont is actually maternally transmitted from louse to louse. This interaction has been co-evolving for the past 25 million years. Our understanding of host-symbiont interactions have actually shaped how host-parasite interactions can be viewed now and years to come. We evaluated the information in light of a modern synthesis of louse evolution. We noted the timing of significant louse-bacterial coevolutionary events, and we suggest potential avenues for further investigation. We mapped out how the coevolution of louse and its endosymbionts has been changing, and the processes and the understandings in the community have been changing over the past years. We conclude by reframing a historic hypothesis of symbiont impact on vector capacity given the information aggregated here. As you can see in the table below, we provide evidence that louse endosymbionts started off as gram-negative bacteria, but subsequently it lost its outer coat. Human head louse, human body louse, chimpanzee louse, gorilla louse, and human pubic louse have been changing in its absence or presence of the outer coat. As you can see in the table, many of the lipid A biosynthesis has been proven to shown as missing. Because the outer coat has been lost over the course of coevolution, we are able to say that we have made a discovery, and we're able to say that there are two different types of human louse endosymbionts. We find evidence that one is associated with lice and that are more efficient vectors of gram-negative pathogens. We provide insight on how endosymbiont and host interactions are likely to drive coevolution and the host response in bacterial pathogens. Because of our research conducted here, we are able to develop a better understanding and more modern understanding of the processes that happens within the human louse microbiome. We're able to further understand how the endosymbiont is transmitted, how the endosymbiont can be classified, and how it not only physically, but procedurally helps the louse in digestion and transmission and reproduction. I would like to thank the Center for Biological Data Science at VCU, my PI, Dr. Brett Boyd, all of my mentors, my lab group, as well as my family for supporting me through this research. It is quite important that this understanding has come to the evolution of where we stand now, and it is quite important that we understand why the human louse microbiome and the endosymbiont interact with each other, how they interact with each other, what this means for the public health, and how this understanding could be further implemented into the medical field and make a socioeconomic difference in the world. This microbial understanding will lead to a macro impact. Thank you so much.